class. Welcome to week 1. This week we're going over chapter 1 and chapter 2. So for the lecture, you're actually going to have 2 parts. I don't think I can edit them together, so you'll just have chapter 1 and chapter 2 separately. So we're going over the science of life, which is kind of an overview of all of the things that we're going to study throughout the semester. And then as the chapters unfold, we'll get into more detail. So if you bought a copy of the physical textbook, that's great. I personally like to have a hard copy in front of me. If not, there is an online book access that comes with the course. You should be able to access that. I'll leave some instructions in an announcement that will open. So if you haven't read that yet, which you should have got that first, you can go back and read it now. So on page 1 of the book, that is where this starts. I do always like to bring attention to the very first box that comes up with our chapter. So there is a blue box here on page 1 that says diversity in science. This is just, it kind of gives you a little bit of cool information that pertains to the chapter ahead. In this case, we're kind of touching on what biology is. So excluding bacteria, our planet is home to a very, very large amount of diversity. So we have over 8.7 million species, including ours, Homo sapiens, that inhabit the globe. And I think sometimes people tend to get caught up on we live in a bubble. We think about ourselves a lot. We don't really think about how many different species there actually are. We know there's a lot of things, but until you put a number to it, it almost is an abstract sort of thing that we don't think about. Biodiversity is very, very important, and because life is so diverse, the science of biology focuses on understanding all aspects of living organisms, exploring life from genes at the molecular level all the way up to large-scale interactions of multiple species in the environments around us, and we do get into that in this chapter. So, with the characteristics of life, like I was just saying, life is very diverse. With shared organization and specific characteristics. So you can see bacteria, we have humans, we have plants, we have fungi, we have insects. There are more things than I can list right now. So there's a lot, and we're very different, and we are made at different levels of what I would just consider complications. Some of us are more simple, and some things are more complicated. So there is an organization in science, and it starts with a cell. That is the smallest, most basic unit of life. We have organisms that are unicellular and multicellular. And then up from there, you have tissues. Those are made up of similar cells. Then you have organs. Those are made up of tissues made from cells. And then you have organ systems, which are groups of tissues working together, or organs working together, and then you have the organism itself. So you have organ systems working together to support an individual. Up from there, we have population. That is where members of similar organisms within a particular area kind of gather. And then we have species. That's where the populations of those similar organisms, they're capable of interbreeding. And then we have community, populations of species that interact within a given area, different from population. And then we have ecosystem, and that's where the communities interact with the physical environment. And then the biosphere, which is the zone of air, land, and water at the surface of the earth where living organisms are found. So all of these things, they might kind of sound like a duh moment when you're thinking about, yes, I know what all of these things are. But we're breaking it down because science isn't just one thing. It's a collection of things, and they can either, depending on which way you look at it, if you're going from biosphere down to atoms, you can make it more complicated to the most simple, but it's important to understand how things break down. So on page 3, there is a figure of the levels of biological organization. Starting at the bottom, you have atoms, and then you have molecules, and then you have cells, tissues, organs, organ systems, organisms, population, species, communities, ecosystem, and biosphere. So all the things that I just said, this is kind of the level, and I like the pictures just because it shows you. Gives you I like the visual representations. I'm kind of a visual learner. Just to be able to see it makes sense. To me, science blows my mind. Biology especially. Just thinking about how things are put together, how they work, what's happening at the cellular level, and the more I learn, the more amazed I am. Now, life requires materials and energy. Life just can't happen without them. You have to have it, and it must be maintained. So we have to have food, of course, or we won't live. That's the building blocks, and that's our energy sources. Energy is the capacity to do work, and then metabolism, that is all of the chemical reactions occurring in the cell. And the ultimate source of energy, of course, for nearly all of life on Earth is the sun. So photosynthesis transforms solar energy into chemical energy for nutrient molecules. So we know what photosynthesis is, but we have a chapter on that later on. The energy and chemical flow between organisms, that defines how ecosystems function. Because energy doesn't cycle, energy could stay in existence without solar energy and producers to convert it. That's a typo, sorry about that. So the dynamics of an ecosystem here. So if you look at the figure, it's on page 4. This is just kind of an example of what chemical cycling is. So you have your solar energy that goes to the producers, emits heat, goes down to the consumers, emitting heat, down to the decomposers, emitting heat, and then that goes back up to producers. So it's this circle of life, if you want to think of it in terms of the Lion King. Everything kind of runs on a wheel, and balance and homeostasis is important. So organisms need to keep themselves stable in regards to temperature, moisture level, acidity, pH, etc. There's many factors with this, but like I just said, homeostasis, that's the internal environment that acts within a set of physiological boundaries. It is the balance. Everything needs to be in balance. Once upon a time, you have probably heard this at some point, but before modern medicine, scientists used to think the body was balanced with something called the 4 humors, which the idea was not correct, obviously, but the concept of balancing pieces of the body and making sure things were at a homeostatic level came into play then. So they kind of had the right idea, even though the details were wrong. Living organisms respond. So if it's alive, there's going to be a response. They find energy or nutrients by interacting with the environment, and the ability to respond often results in movement of some sort. Living organisms also reproduce and they develop. Every living thing can reproduce or make another organism like itself. It can make a copy or grow a copy. So bacteria and other single-celled organisms, they simply split in 2. In multicellular organisms, the reproductive process usually begins with the union of an egg and sperm that produces an embryo. Then the embryo grows with the genes inherited from the parents and according to the rules from those, and then in all organisms, the genes are made of DNA, and DNA is the blueprint. Breaking it down, which these are all things that you have heard. We know how children are made in most species. But that is some of the ways that you can define a living organism. Reproduction is a characteristic of life. So whether it is single-celled or multicellular, all organisms reproduce. You'll either have an offspring that receives a copy of the parent's DNA and therefore a copy of their parent's genes. So another characteristic is reproduction. Living organisms also have adaptations. These are modifications that organisms will make to help them survive or be better suited to their way of life. For example, some hawks will catch fish, but others are better at catching rabbits. These are adaptations for flight and for catching prey. Depending on the bird, they will have different flight patterns or flight characteristics, and that usually helps them in the way that they hunt or what they hunt and where they hunt, so they can survive. It is essentially a survival of the fittest when it comes to adaptations in life. So humans who live at extreme elevations, for example, they have an adaptation that reduces the amount of hemoglobin in the blood. And just to sort of summarize what that means, you have more red blood cells in your body to carry oxygen more efficiently because the higher altitude makes it a little bit more difficult to get oxygen to your body because of the normal amount of hemoglobin helps with that. Just FYI. Evolution, this is a core concept of biology. This is the process by which populations accumulate adaptations over time to become more suited to their environments. This is what I was saying a second ago, survival of the fittest. Due to evolution, people adapt to their environments and they either survive or they don't. And this isn't just people, this is with every species. This explains the unity and the diversity of life. Evolutionary trees trace the ancestors of life on Earth to be a common ancestor. So you can take things way back. And we do have chapters on evolution later, and we'll dive into more details on that. But here's a figure of an evolutionary tree, just kind of a basic figure. And you can see on the left underneath it says the first ancestral cell. This is on page 7. So you have bacteria, and then it evolved into archaea, into eukarya, plants, fungi, and animals from there. So you can just kind of see they trace things back to a single key linking ancestor. We have natural selection. This was something that Charles Darwin and Alfred Russel Wallace both independently came to the conclusion that evolution occurs by a process called natural selection. Darwin wrote on the origin of species. It's a book he wrote as he was on a boat kind of observing the way that things were on different islands. If you haven't read on the origin of species, you can get it off of Amazon for about in between $10 and $15. It's an older book, so some of it's a little difficult to read, but it's sort of a staple in biology. So if you find these sorts of things interesting, I do recommend reading it at least once, because he's spot on with a lot of his thought processes, and it really opened the doors for science, for biology. And like I said, it's a core concept of biology. It explains so many different types of observations in every field of biology. And we can see natural selection happening today as it unfolds before us. We can mark it down. Environments change a lot. That's due to the influence of living or non-living factors. And natural selection is the process that results in a population adapted to the environment. So some individuals or populations might have certain adaptations that make them better suited to a new environment. Others won't. Those individuals that are better suited to the new environment will then live longer and produce more offspring. And those adaptations that result in higher reproductive success, that will then increase in frequency from one generation to the next, and that is evolution. The things that allow you to survive write themselves into your genes, and they stay. They'll sometimes stay so long that they end up a permanent fixation in the DNA. That's another subject and concept altogether, but they can write themselves in and they'll be permanent. The process of evolution can be summed up as what they call descent with modification. Hawaiian honey creepers is a good example of this. So these all evolved from one species of finch. The assortment of bill types, they have changed depending on what types of food the finches eat. But some of them still share certain characteristics from the common finch ancestor. So they have body shape, nesting behavior, other things like that. But here's a picture of that. If you want a closer view, or if you want to kind of like stop and zoom in, or if you want to screenshot it and zoom in, you can. But you can just take a look and see that the beak shapes are completely different. It doesn't tell you here what they eat, but there is a specific family that has lived on a certain island for about 20 years, and they have studied a similar bird and watched, like, during wet years what happens to their beaks and the offspring that happen after that, what happens during dry years, what if they eat harder shelled nuts or things like that. Their beaks are a different shape, whether they eat softer things. So you can see, it's wild, just in beak shape alone, and that's just one thing. Now we do have taxonomy. This is the discipline of naming and classifying organisms according to certain rules. So of course we have rules. The systematics that we use classifies organisms according to presumed evolutionary relationships. You've probably heard these at some point. I will probably ask you a question about this. Now, the way that I learned this, you can put an acronym to it if you want to. Supergroup, the second one, we'll talk about that here in a second. This is a relatively new addition, but I used to have the sentence, do kings play chess or fight giant squids? I like that acronym because the sentence itself makes sense, and that just means it's domain, kingdom, phylum, class, order, family, genus, species. And I'll get into that in just a second. Now, if you want to maybe change the acronym, we could say, do some kings play chess or fight giant squids, just to kind of still make it a sentence. It's classified into 3 domains. The most inclusive and most general is domain archaea, is the domain. So we have archaea, that's like for prokaryotes, they're unicellular, they don't have membrane-bound nucleus, and they are what is considered the representative of the first cells on Earth. Then we have domain bacteria, also prokaryote, unicellular. They also don't have a membrane-bound nucleus, and they are found almost everywhere. They are what is considered ubiquitous. And then domain eukarya, hello, that's where we live. So eukaryotes, we are unicellular or multicellular, and we have a membrane-bound nucleus. So you can see here for archaea, this is sort of like what I think of this almost as like ancient. It's old. They're capable of living in extreme environments. So you have some that are salt lovers. They call those halophiles. You have, well, you can classify them as a whole as an extremophile. There are others that tolerate salt, some that tolerate heat, some that tolerate extreme cold. Some tolerate pressure, that sort of stuff. So there are a lot. You can find these living in many, many places. Bacteria, they're structurally simple but metabolically diverse. For example, whenever I worked on my research, one of the tests that I did were sugar fermentation profiles. And what that means in simple terms is we tried to find out what they ate and where they got their energy from. So I did tests with sucrose, maltose, glucose, and I don't remember the last one, but I did 4 different sugars and a bunch of other characterization tests. But what I was trying to find out is what these bacteria used for energy, and if they didn't have that as an energy source, would they use something else? And the answer is that about 90% of them could use any of the 4 things. They had a preference. You know, some of them would take sucrose over glucose or what have you, but many of them, if they didn't have one, would convert to the other. And the difference in metabolic pathways down the line makes a difference in what antibiotic you choose to treat it with, because some will target a specific pathway and some will target a different one. So they are very metabolically diverse, which I think contributes to their ability to survive and multiply the way that they do. Now into the supergroups and kingdoms. So historically, the domain eukarya was divided into 4 kingdoms, and then improved techniques in analyzing DNA has suggested that not all of the protists share the same evolutionary lineage. So evolution of the eukaryotes has occurred along several different paths. A taxonomic group now called supergroup was developed to explain these evolutionary relationships, and there are currently 6 supergroups in the domain eukarya. And they are listed here. You can actually see this list on page 10. So we have Archaeplastida, Chromalveolata, Excavata, Rosaria, Amoebozoa, and Epistoconta. So these are, you can see what's in them. You have plants in the first, flagellates, diatoms, golden algae, so on and so forth. So it just kind of shows you the breakdown. I'm not going to test you on this portion, but it is interesting to see because they've added this in recently. So taxonomic names and things like that, they change all the time to better classify. So the traditional kingdom level classification of domain eukarya, that's still widely used. 4 eukaryotic kingdoms are recognized. We have protists, that's a diverse group ranging from single-celled to multicellular forms. Some use photosynthesis to manufacture food, and some have to acquire their own food, including algae, protozoans, and water moles. The other three kingdoms, those evolve from the protists. So we have plants, kingdom Plantae. They're multicellular photosynthetic organisms, azaleas, zinnias, pines, etc., etc. Then we have the fungi, kingdom Fungi. They are the familiar molds and mushrooms that decompose dead organisms. We kind of know what the fungi do. If you're interested, there is a show on Netflix, I think, or it used to be called Fantastic Fungi. Or fungi. It's a really good, it's kind of slow, very informative. If you like facts, neat facts about things, I thought it was interesting. They're way more complicated than I had thought before, so I liked it, but I'm also a nerd. Kingdom Animalia are multicellular organisms that ingest and process their food, so that's where we live, and we are also in the category with jaguars, hawks, etc., etc., etc. So here are the 4 eukaryotic kingdoms. That's on page 11. Now each organism has what they call a binomial name. That means there's two names. For example, the garden pea is Pisum sativum. The first word is the genus, and the second word is specific to the species. Homo sapiens. For example, Homo is the genus, sapien is the species. For the gray bat, that's Myotis crescens. Myotis is the genus, crescens is the species. There are many different types of Myotis bats, but the species are different, so that's important. It's universally used by scientists to avoid confusion of the common names. Now we get into the scientific method. So because biology is the scientific study of life, we have to have a way to study that with rules. So the first thing you do in science is you start with an observation. That's how the scientific method begins. You take advantage on knowledge and experience of other science. Even like whenever I was doing my research, I did what they consider a novel study, so it was new, but to formulate my experimental design, I read probably 30 different papers from people who had done similar studies using different parts of the bat. I used their gut microbiome from guano, but a lot of other people would take tissue samples. So they would actually take like tissues from their throat, or they would do urine swabs or pieces of their wings, things like that. So they did similar studies, a lot of virome studies. So they didn't really study the bacteria, they studied like the viral side, because it's known that bats carry diseases. So especially during the COVID pandemic, the study on bat diseases was very popular, which is kind of how I happened into my study and got the approval for it. It was a subject people were interested in. Once you have an observation, you develop a hypothesis. So, for example, when I did my study, I was talking with another grad student at the time, and she was telling me about white nose fungus, which is Pseudogymnoascus destructans, genus and species, and she found evidence of white nose scarring in these gray bats. Now, usually white nose fungus is very deadly, especially to tricolored bats or little brown bats. It gets into their colony, and because they live so close together, it spreads like wildfire. So it's just decimating bat populations. But these guys had survived. And I was so curious. How did they survive? Was it just their size, or did they have something going on genetically? And so that sparked an interest. I went and talked to my advisor, and I was like, hey, I'm really curious about this. Haley says they have some guano. Can I test it? Can we design a way to test it? And so it started with my observation. They survived white nose fungus. And then, of course, I was like, why? I developed a hypothesis. I thought that female and male bats would carry different bacteria, and I thought that it was something genetic that allowed their survival. Turns out it was mostly their size, but they're a very resilient species in a way. but it's more their size than anything. But that was where my question started. So then we had to develop a hypothesis. So we had to use inductive reasoning and create a way of thinking to be able to test these facts and put them together. So a hypothesis is your possible explanation. It's what you think is going to happen. So my hypothesis for my thesis was I predicted that male and female bats would house different bacteria in different amounts. But I also just kind of had a general question on what bacteria did they hold, just period. What did they have? And was it dangerous or not? And then we had to find a way to test it. So you have to consider the questions that you can test. If you can't test it, it means nothing. Because in science, we love statistics, and we have to find a way to get the statistics, and the only way to do that is to have a question that you can test for. So this is a good flow diagram for the scientific method. I'm not going to go go through all of them, but really you have your observation, your hypothesis, and then you come up with your experimental design, and then you do your test, and then you either reject or accept your hypothesis, and then you have your conclusion. So if you want to break it down simply, we could say observation, hypothesis, experimentation, conclusion, results. Once you have your results in science, it's kind of a known thing. You share your information. You publish it. You have it peer-reviewed. So some of the really important things in science come down to the peer review process. It's where other scientists look at your work and they try to find the weak spots. Some people struggle with this because constructive criticism can be difficult to hear, especially if they're tearing your experiment apart. I was scared to death whenever I sent in my thesis for my first round of edits. But once you get the first round out, it's really nothing after that. I would rather get edits back than me sending it off and someone say, oh yeah, this is great, and then I publish it and then I realize that I've embarrassed myself. Yeah. Because what if you made a big mistake? So, you make a prediction, you perform your experiments, and then those experiments further the observations and test your hypotheses. A good experimental design, all conditions are constant except for one experimental variable, so that's your control. You have to have a control in an experiment to say, look, you can't just have numbers and then have nothing to compare it to, so the control is the comparison. Sometimes data might suggest a correlation, but correlation does not mean causation. One good example of this was when vaccines really went on the rise. A lot of people noticed that autism rates were also on the rise. And it was stated that vaccines cause autism. Now, just because those two correlations happen doesn't mean that one causes the other. In fact, they've done many, many tests since and found it not to be true. But that can lead you into dangerous territory. And that's another reason why the scientific method and proper experimentation is important. So then you have to get around that. So scientists are naturally skeptics. I think being a skeptic is a good way to live life. Do your own research. I always encourage people, don't take my word for it. I could be wrong. Look it up for yourself. Come up with your own conclusion. So then you develop a conclusion, and then you have to say if the hypothesis was supported or not. One other thing I'd like to mention is that in experiments, the observations must be repeatable. Repetition and randomization are extremely important in science. You cannot allow bias to come into your experiment at all. And so that's why we do something called a double-blind study, which I'll go into in just a second. So with theory is different than hypothesis. So theory comes to be whenever you have a lot of supporting documents. So the ultimate goal of science is to understand the natural world and accepted explanations for how it works. Cell theory or gene theory. Theory of evolution is considered a unifying concept in biology. We have a lot of numbers to back this. Some biologists even refer to the principle or law of evolution because they have about 100 years of support by observations and experiments. So they have a lot of backing for this. You have to have a lot. to consider something a theory. So a controlled study is the best way to do an experiment, and an example of this is actually on page 14. I have a picture I can show you here. So the hypothesis is that antibiotic B is better than antibiotic A in current use for the treatment of an ulcer. So there's 3 experimental groups. This reduces possible variations by randomly dividing the large group. So you have repetition and randomization. And then a control group receives the placebo. So out of the 3 groups, one is a placebo, that's your control. You have test group 1 and test group 2. Whenever I did my experiment for testing female and male bats, their guano, I had, I think, 6 samples of each. So I mixed it all together, and I split it up, and I did the test on the males 6 times. And then I put all the numbers together. So that way I was sure of the numbers that I was getting. So here's a 1, 2, and 3 group with the control, antibiotic A and antibiotic B. And then each subject was examined for the presence of ulcers. And then you can see in the chart, so 60% of test group 1 were cured and 80% of test group 2 were cured. So it seems like antibiotic B works better. Now, a double-blind study was done. So the technician doesn't know what group the patient they were examining was in. They're not told this information. So not only do you have a control and randomization, but you got somebody running the numbers that has no idea who's in what group, and that keeps bias out of it. So that helps give a solid conclusion. Now we do have some challenges in science. So technology is a big challenge. That's the application of scientific knowledge for a practical purpose. Technology allows us to do a lot of really cool things. It allows us to alter organisms and ecosystems to address the needs of human society in many, many different ways. But despite our scientific and technological advances, humans face a lot of severe challenges. Climate change is primarily due to an imbalance in the chemical cycling of the element carbon. Normally that's cycled in the ecosystem, but due to human activity, we got more carbon dioxide being released than can be removed by ecosystems. So in 1850, the atmospheric carbon dioxide levels were about 280 parts per million, but today it's over 400. So that's a monitorable increase. And here is a visual representation. You can clearly see it's not going in a good direction. But we also have habitat loss, so biodiversity is important, and we need it for variation in life. We have to have diversity in order for things to survive. So like I said, there's around an 8.7 million species estimation, that's excluding bacteria, and there's around 2.3 million classified, so there's even more than we actually have classified. Extinction, that's the death of an entire species or a taxonomical group. So about 38% of all species are in danger of extinction by the end of this century. There are things that go extinct every single year, sadly. The present rate of extinction approaching levels of the 5 mass extinctions in Earth's history. So it's getting dangerous. Extinction affects biodiversity, and then the ecosystems, they can't thrive without biodiversity. So this is one of those snowball effects. We also have emerging diseases. Those are the ones that are relatively new to human. For example, COVID. But we also have the avian flu, the swine flu, SARS, and then MERS, and then, of course, SARS-CoV-2. And like I was saying, emerging diseases, this is something that they were looking at bats for, because bats are disease vectors, and they can carry things that are considered zoonotic, which is where they can jump from species to species, and that's how people get sick. And they carry a lot of different diseases, so we would consider that emerging diseases, because they can result from new or increased exposure to animals or insect populations that act as vectors. And then because of globalization, and the way that people travel from country to country and move around all the time, that increases worldwide transmission. And the Ebola outbreak in 2014 and 15 is a perfect example of that. And that concludes this chapter. I'm going to go ahead and stop this here, and then we'll jump on over to chapter 2 and continue.